imply that the carboxyl group, the C double bond O group, is on the first carbon in the long carbohydrate chain. Then the ketones imply that it can be on any other carbon, other than the first carbon. It can be on carbon number 2, carbon number 3, carbon number 4. So long as it's not on carbon number 1, it falls in the functional group of ketones. The other classification of carbohydrates can be based on size, where we have monosaccharides, which are single monomer units of carbohydrates. You have disaccharides, when you have two carbohydrates bound together. We have oligosaccharides, when you have carbohydrates of about 10 in one chain or other arrangements. Then we have polysaccharides. This is a large arrangement of carbohydrates. So that's the general introduction I can give on carbohydrates based on their classifications and its empirical formula. So carbohydrates have numerous functions in our body. In the layman's language, they produce energy. So certain carbohydrates contain sugar and starch. And these carbohydrates yield energy through oxidation, which we'll cover in the near lectures above. So oxidation occurs, and these carbohydrates are able to produce energy. Equally, insoluble carbohydrate polymers can serve as structure or protective elements in cell walls of bacteria and connective tissue in cells. An example I can give are the nitroglycosaminoglycans. Yes, the nitrogen glycosaminoglycans. These are an example of carbohydrates which are insoluble and tend to have a protective or structure function in cell walls of bacteria. I'm sure you'll find it should be N-acetylglycosaminoglycans. Yes, N-acetylglycosaminoglycans. These are a class of carbohydrates which are found in cell walls of bacteria and connective tissue of animal cells that serve for structure and protective functions. So other carbohydrate polymers can lubricate joints to prevent friction when joints are moving. Then specific carbohydrates contain molecules that act in cell-to-cell recognition. For those who remember the last lecture we had, that should have been physiology, and I was helping out the physiology department. We were talking about when you're looking at the cell wall, remember finding some glycoproteins which help in cell-to-cell recognition and how cells recognize themselves and differentiate themselves from other cells. Equally, certain carbohydrates help in adhesion, meaning joining to other molecules, in blood clotting, the immune response, and wound healing. These are some of the functions that carbohydrates serve. Okay. So now, so far, are there any questions just on the introduction and the functions of the carbohydrates before we start moving and start looking at each carbohydrate from the monosaccharides, disaccharides, oligosaccharides, and the polysaccharides? Are there any questions before we proceed? Okay. I will take the silence as there are no questions, and we can proceed to the next part we'll be looking at. So we'll first look at monosaccharides. These are the simple sugars. This can be trioses, when then tetroses, pentoses, hexoses, heptoses, so on and so forth. Now, this classification of monosaccharides solely depends on the number of carbon atoms in the backbone of the chain of that monosaccharide. If there are three carbon, there are trioses, 4 tetroses, 5 pentoses, 6 hexoses, and so on and so forth. So depending on the number of carbons, they can be classified as such. Monosaccharides can equally be classified depending on the functional group they have, either an aldehyde or a ketone. So an example I can give is this one, D-glucose. So D-glucose is an aldehyde. Now it's an aldehyde due to the position of the carboxyl group. This carboxyl, the carbon oxygen double bond group, is on the first carbon. As a result, this is an aldehyde because the functional group here is COH. This functional group, I hope you can see where my cursor is. This functional group showing here is an aldehyde functional group. You'll come across this in more detail when you look at it in your organic chemistry. Then the other functional group on the other side is an ketone group. When you have a C double bond and this classification you can give to monosaccharides as either aldehydes or ketones, and based on the number of carbon atoms in the chain. And look at this one here, it's got 1, 2, 3, 4, 5, 6. This group, and it's a triose in that it's got 3 carbon atoms. So these two carbohydrates, monosaccharides given, have a very important physiologic significance. Now, when you begin to look at glycolysis, which is the breakdown of simple sugars in order to produce pyruvate and energy, these two carbohydrates you're seeing, you'll find them in the 5th step of glycolysis. Glycolysis is a 10-stage process in which carbohydrates are glucose is broken down into pyruvate and energy is produced. So these two are carbon atoms found in that glycolysis change and are very important in the formation of pyruvate. So these monosaccharides are usually colorless, water soluble, and crystalline solids, and they have a sweet taste. So keep these in mind. Glyceraldehyde and dihydroxyacetone. These are important monosaccharides. So keep them in mind. So all these are examples of aldoses. So aldoses, which simply entails that the first carbon is on position 1 group, sorry. So when you have the carboxyl group on position 1, so excuse me, just give me a minute. Something has come. Just give me 1 minute, sorry. Give me 1 minute, 1 minute.
We can get
Yes,
you.
sir.
Okay, apologies for that. So we proceed. So all these in this are all simply examples of example of a 3-carbon aldehyde, aldose or aldehyde is D-glyceraldehyde. An example of 4, we have D-erythros. of sugars, which are equally important. So majority of derivatives that can be modified. So glucose is a hexose, and majority of them can be modified to make with the first one. So the first aldonic acid is formed. Now, when you look at this carbon here, D-glucose, for example, it has carbon 1, 2, 3, 4, 5, 6. Now, if the carbon, if there's oxidation of this aldehyde group, as you can see here, D-glucose has an aldehyde group. If this aldehyde group is oxidized at carbon 1, the an aldonic acid. An example of D-glucose, once it's oxidized, what you form. So these as the first derivative we'll look at. So now these acids have a broad range of applications in the food industry. They act as and in cosmetics as antioxidants. So the first sugar derivative we've looked at is aldonic acid. So aldonic acid is formed when there's the aldehyde group at carbon 1 of an hexose, in this case glucose, then the compound formed is a gluconic acid. So an example given here, glucose is oxidized. Hope we're together. So the second one you can form, the second derivative is uronic acid. Now, uronic acid is different in that it's formed by the oxidation of the hydroxyl group at carbon number 6. Carbon number 6 of the hexose sugar, of which this case is an aldose sugar because we have that aldehyde group there. So uronic acids can assume a pyranose, furanose, or linear type of conformations that they can have. It can either be a linear or a furanose forming a ring with 5 carbon in it, or a pyranose, depending on that physiological need. So the example given of a uronic acid is when D-glucose is oxidized by the oxidation of the hydroxyl group to form D-glucuronic acid. So remember, on the first one we have gluconic acid, then on the second one we have glucuronic acid. So this is the second derivative of sugars. So these normally are steroids, and they appear in urine of animals as normal metabolic products. So D-glucuronic acid is also a major component of connective tissue polysaccharides. So we have looked at 2 derivatives of sugars. Now, the third one is aldaric acids. So now, the combination of the first one and the second one, when an aldehyde group is oxidized at both the first carbon atom and at the 6th carbon atom, when the aldehyde and the hydroxyl groups are both oxidized to form carboxyl group, the COOH, sorry. Now, these aldaric acids don't form cyclic structures. So aldaric acid, yeah. So an example, when oxidized both at carbon 1 and carbon 6, where we have at carbon 1, we have the oxidation of the aldehyde group and carbon hydroxyl group, carboxyl groups at both carbon 1 and carbon 2. What you form is glucaric acid. So we looked at glucanic acid, glucuronic acid, and glucaric acid. These are 3 derivatives of sugars we have looked at so far. The next derivative we can look at is alditors. Now, the first 3 were formed by oxidation, but alditors are formed by the reduction of an aldose to a polyhydroxyl alcohol. So they're forming an alcohol. So an example is given here when glucose is reduced to sorbitol. So we have this glucose here. This is a glucose molecule. We have an aldehyde group at carbon 1 there. Of course, we can automatically, when you have the bend there, we know there's a carbon atom there and a hydrogen atom. That's said from organic chemistry, from chemistry 1010. So when this glucose is reduced by an enzyme, in this case, the aldose reductase, which catalyzes the reaction. So this NADPH is a hydrogen donor. So it donates its hydrogen to glucose, thereby leading to the formation of sorbitol. And this NADP plus becomes positively charged because it's lost its hydrogen. So sorbitol is formed by the reduction of the aldose to polyhydroxy alcohols. So these are alditors. The use of alditors are mainly as in the food industry or in chemical reactions molecule. An example is glucose when it's converted to glucosamine. So we have this glucose here in its cyclic structure. This is glucose here. So now this glucose, first of all, at position 2, is replaced by an amine group. Thereafter, it's also replaced by an amino group. This functional group was seeing the NH, C carbon double bond O CH3. This is an amino group. So when you have an amino group added to a sugar, what is formed is an amino sugar. In this example, we have glucose being converted to glucosamine. Glucosamine, sorry. Now, the amino group is nearly always condensed with acetate to form N-acetylglucosamine, which is the part of the structure of polymers, which is found in bacterial cell walls, as we're discussing in the functions of carbohydrates. We have the N-acetylglucosamine, which is the part of bacterial cell wall, which gives structure and structure support that bacterial cell wall as a function of the carbohydrate. The other ones we can look at are sialic acids. Now, these sialic acids are 9-carbon sugar derivatives that are present at terminal residues of glycans and glucoconjugates. So now, an example which is given is when N-acetyluramic acid, N-uracetic, that's an example of a salic acid. Now, N-acetyl monosamine reacts with phosphonopyruvate, PPP, phosphoenopyruvate, and they form N-acetyluramic acid. So an example which is given is this chemical compound we're looking at. So this is N-acetyluramic acid. So don't really worry much on the structure of this. You will not really be examined or asked to draw the structure of N-acetyluramic acid, but you just have to know that it's an example of a sugar derivative from salic acids. So these salic acids have functions such as in cellular communication and stabilizing cellular membranes. These salic acids also help in the binding and transporting of ions and sugars, equally help in the formation of proteins such as enzymes, and equally protect molecules and cells from attack by proteases and glucosides. Before we move on to disaccharides, are there any questions on monosaccharides and their derivatives? Any questions on monosaccharides and their derivatives?
You are saying we shouldn't worry about the structures.
Yes, you shouldn't really worry much on the chemical structures of those compounds because the biochemistry we're doing is medical biochemistry. So I'm mainly focusing on the medical aspect of biochemistry, the functions of those enzymes. Maybe if those enzymes are missing, what happens when those carbohydrates are missing, what's the effect on the body. So you won't really be doing the biochemistry as the biochemist would be doing it mainly on the medical field. So these structures are not mainly there for you just to have the basic understanding and the knowledge of what they look like. Yeah.
Okay, thank you.
Most welcome. Any other question, or we can proceed to the disaccharides? Okay, I'm guessing you can proceed. So no more questions. So disaccharides are formed by a condensation reaction of two monosaccharide units. So when you look at disaccharides, I'm sure you did this in biology last year. Yeah, you guys, I'm sure you did this already in biology last year. When you have two disaccharides, a condensation reaction occurs. Why it's called a condensation reaction is because the water molecule is given out, and as a result, they are joined. So examples of disaccharides are maltose, lactose, and sucrose, of which you know that maltose is formed when two glucose molecules are condensed, lactose when galactose and glucose are fused together, they form lactose. Then sucrose is formed by fructose and glucose. These are some of the disaccharides. So two monosaccharides joint covalently by a O glycosidic bond. So the bond that joins this bond here, that joins two monosaccharides to form a disaccharide, is the O glycosidic bond. So we've already talked about most of these disaccharides and their functions. Yeah, we have talked already about the maltose from two sugar residues, lactase, sucrose, all those are simple sugar derivatives. Okay, so not much really can be talked about when it comes to the disaccharides, but one thing you have to note that there are some people who have a deficiency in lactase. So lactase is the enzyme that breaks down lactose into D-glucose and galactose. Yeah. So now these normally leads to malabsorption, leading to diarrhea and flatulence. Yeah. So those who we talk about are lactose intolerant, as we commonly call them, lactose intolerant. Simply means they cannot process lactose simply because they lack the enzyme lactase, which breaks lactose into D-glucose and D-galactose. As a result, they have diarrhea and flatulence when they take products containing lactose. So if lactose accumulates in the lumen of the small intestine, so as it accumulates, that causes a discomfort that most people who are lactose intolerant feel when they take milk or dairy products. Yeah, that's one note you have to take note of when looking at disaccharide, on lactose or lactase deficiency. Yeah. And the next thing you can look at are polysaccharides. Now, most carbohydrates found in nature occur as polysaccharides. That's the most common way you find them. And they're also called glycans. So now, we have two types of polysaccharides. We have homopolysaccharides and heteropolysaccharides. Homosaccharides simply means that they contain only a single type of monomer. An example of a homosaccharide is a polysaccharide that is made only out of glucose molecules only, just glucose, forming a polysaccharide. That's an example of a homosaccharide. Whereas the heterosaccharide is when you have two different monomers, an example of maybe galactose and glucose forming a polysaccharide. As a result, they form a heteropolysaccharide. So, other homosaccharides include cellulose chitin, for example, chitin, and the cells are structure elements in plant cell walls and animal exoskeletons, whereas heteropolysaccharides provide extracellular support for organisms of all kingdoms. So this is just a picture view of a homopolysaccharide. It can be in a linear. Well, notice that even just from the color coding, it's made simply from one monomer repeating unit. Then we have this, still a homopolysaccharide. The only difference is that it's a branched homopolysaccharide. The other one we can look at is the heteropolysaccharide, which has two or multiple different monomers. As you can see, for the linear one, we have only two colors, which signify that there are two different monomer subunits making up that polysaccharide. Then we have the branched one, which has 3 different monosaccharides. So we have homopolysaccharides made from 1 repeating unit, as well as heteropolysaccharides made from multiple repeating units. That's the 2 classifications you can find polysaccharides in. So the first thing we look at are the homopolysaccharides. Now, these are most important because they are storage polysaccharides in starch. They are important for storage. So homopolysaccharides important for storage are 2. We have starch in plant cells and glycogen in animal cells. That's the storage form of glucose in the body, or the storage form polysaccharides in the body. So remember, starch, plant cells, glycogen, animal cells. So both polysaccharides occur intracellular as large clusters or in granules. So they can be clustered together, or they can just be in granules. So starch and glycogen molecules are heavily hydrated because they have many exposed hydroxyl groups available through hydrogen bond with water. So that's why they're called heavily hydrated, because they have so many hydroxyl groups that can hydrogen bond with water. That's on the homopolysaccharides. So we'll start by looking at starch. Now, starch is found in tubers such as potatoes and in seeds. So if you remember from your biology, starch is in 2 forms or 2 types. We have amylose and amylopectin. So amylose is the linear form of starch, which is long and unbranched, formed of D-glucose residues and connected by alpha 1,3 linkages. Simply means the carbon 1 of one glucose molecule to carbon 4 of the next glucose molecule. That's how amylose is starch, and it's unbranched. Simply means it's just one straight line, so to say. Now, the other form of starch is the amylopectin, which is highly branched. So since it's branched, so it's got 2 joinings. It's got the alpha 1,4 for the linear and the alpha 1,6 linkages for the branches. And each branching occurs every 24 to 30 residue. So when a amylopectin molecule starts, from 1 to 24, there'll be a branch. Another 1 to 24 or 25, but within the range of 25 and 30, there'll be branches that will be occurring. So both amylose and amylopectin are hydrolyzed by alpha amylase. So alpha amylase is the enzyme that hydrolyzes the internal alpha 1,4 linkages to yield maltose, which are 2 glucose residues. In the alpha 1,4 linkage, the maltose, which are 3 glucose residues, so on and so forth. So the amylase enzyme works both on amylose and amylopectin to break them down into polysaccharides maltose. Yeah. So these are simply pictorial views showing you the amylose, which is simply linear, and you can see the alpha 1,4 linkage joining this linear structure of amylose. Then we have amylopectin here. So amylopectin clearly has this is an alpha 1,4 linkage, as well as a 1,6 at its branch point. So those are the 2. This is a pictorial view showing the 2 forms of starch, being the amylose and the amylopectin. Okay, the next thing we'll look at is glycogen. Now, remember, starch is the main storage unit of plants, while animals use glycogen to store their carbohydrates. So it is a polymer and has alpha 1,4 and alpha 1,6 linkages. So glycogen is synonymous or similar to amylopectin. The only difference is that it's extensively branched. Remember, in the amylose, the branching was taking place every 24 to 30 residues, But for glycogen, the branching is taking place every 8 to 12 residues. Therefore, glycogen is more compact as compared to starch. So glycogen is abundant in liver and in skeletal muscle. So when there is need for formation of energy, or maybe when the body is running low on carbohydrates, the glycogen is mobilized in the liver or in the skeletal muscles, turned back to glucose and used to form energy. Yeah. So in hepatocytes, so hepatocytes are simply cells that are found in the liver. Yeah, hepatocytes, you'll come across them in your anatomy. Now, they contain glycogen. Glycogen is found in large granules. So when you find hepatocyte, you find that there are a number of large granules which contain glycogen. And glycogen granules contain the enzyme responsible for the synthesis and the degradation of glycogen when there is need for it to be converted back to glucose to yield energy for the body. This, yeah, that's in the starch and the glycogen. This also is the pictorial view, the picture showing the arrangement of glycogen. We have the linear here, the alpha 1,4 branch points and the alpha 1,6 branch points when making the branching. That's the 2 amylose and glucose. So now, the next thing we'll look at are the heteropolysaccharides. So we're done looking at the homopolysaccharides. So the next thing we'll look at are the heteropolysaccharides, beginning with the peptidoglycans. Now, the rigid component of the bacterial cell wall is a heteropolymer with alternating beta 1,4 linkages between N-acetylglucosamine and N-acetylmuramic acid residues, as seen in the structure here. So we have these beta 1,4 linkages here. So we have this N-acetylglucosamine here. So it's different repeating units. So we have this in, should be red, if I'm not mistaken. Yes, that's N-acetylglucosamine. Then the next one is N-acetylmuramic acid. So these two monomers are repeating units formed in the heteropolysaccharide, which is used and found in bacterial cell walls to give them their structure, for structure support. These are peptidoglycans. So we've already talked about that. The next ones we can talk about, so we've talked about the peptidoglycans. The next one we can look at are the glycosaminoglycans. Now, glycosaminoglycans are a family of linear polymers composed of repeating disaccharide units. So, one of the two monosaccharides is always either N-acetylglucosamine or N-acetylgalactosamine, and the other, in most cases, is uronic acid. So that's how glycosaminoglycans are formed, by repeating units of disaccharides. So the extracellular matrix, I'm sure you've looked about it, the ECM, is composed of interlocking meshwork of heteropolysaccharides, the glycosaminoglycans, and fibrous proteins such as collagen, elastin, and fibronectin. So these glycosaminoglycans will be found in the extracellular matrix. So these are some of the other examples you can find. We have the hyaluronates, we have the chondroitin sulfate, dermatin sulfate, keratin sulfate, and heparin. All these are found in the extracellular matrix. So for the hyaluronates, they are found in the ECF of cartilage and tendons. For chondroitin sulfate, it gives tensile strength to cartilage, tendons, ligaments, and walls of the aorta. So dermatin sulfate contributes to pliability of the skin and is also present in blood vessels and heart valves. Keratin sulfate, keratin is found mainly, remember, in nails, hooves, claws, and so on. These are found in those structures. Then heparin is normally an anticoagulant, so it's normally found in blood to prevent blood from naturally coagulating. That's on these other glycosaminoglycans. Then we have glycoconjugates. Glycoconjugates are simply structures formed when you mix carbohydrates with other macromolecules. An example are proteoglycans when you mix proteins and glycogens. Glycoproteins, same thing. Glycolipids when you have glycogen, which are, of course, carbohydrates and lipids. So these carbohydrates can be covalently linked to other biomolecules like proteins and lipids, as we've already discussed, as examples of the proteoglycans, glycoproteins, and glycolipids. So the first one, if you're looking at, are the proteoglycans. Now, proteoglycans are macromolecules on the surface of the ECM, extracellular matrix, in which one or more glycosaminoglycan chains are joined covalently to the membrane protein and secreted proteins. Now, the glycosaminoglycans commonly form a greater part of the proteoglycan molecule. Proteoglycans are mainly found in connective tissue. An example is cartilage. So that's where you find these proteoglycans, which are simply a combination of carbohydrates and proteins. The other ones we can look at are glycoproteins, in which carbohydrates or oligosaccharides are covalently joined to proteins. These can be found on the outer surface of the plasma membrane in the ECM and in blood. So now, they're also found in specific organelles such as Golgi complexes, secretory granules, and lysosomes. These are other places where you can find these glycoproteins. So glycophorin is a major membrane glycoprotein in human erythrocytes. That's just an example of a glycoprotein. The glycophorin is another example of a glycoprotein. Other examples we can look at are glycolipids, simply glycogen or glucose molecules or carbohydrates joined with lipids. So the gangliosides are membrane lipids of eukaryotic cells made up of sialic acid and glycosphingolipids. All these are just simply giving us examples of glycolipids. They act as specific receptors. So the oligosaccharide groups on gangliosides help in cell recognition and cell-to-cell communication, as we already looked at the glycoproteins. Also, so these glycosides equally help in cell-to-cell recognition. The other thing we can look up are the lipopolysaccharides. Now, these lipopolysaccharides are normally found on gram-negative bacteria. You look at this mainly when you start talking about your introduction to your microbiology. You'll find the gram-positive and the gram-negative bacteria and how they're classified in that way and why, and they're classified in those different classifications. Mainly, the lipopolysaccharides found have a role to play, but I'm sure you cover that in your microbiology. But the important thing that we have to know for us in medical biochemistry is that lipopolysaccharides are an example of glycolipids and normally are found on gram-negative bacteria. I think for today, we'll end here on carbohydrates. Then the next time we'll meet, we'll finish up on the qualitative and quantitative analysis of these carbohydrates. Are there any questions?